Perfect. Okay, well, today's presentation is going to focus on managing multi-site research collaboration and how Flywheel can help with these common challenges. So research is hard enough. Multi-site research can introduce some additional challenges. So some of the things we're going to talk about today are, just first off, how you can share data and analyses effectively and cost effectively. Ease of use is obviously a critical factor in being able to do that across sites with diverse users and maybe coming from different IT settings and so on. So we're going to talk a little bit about that. Security and access across sites is another common challenge. Data collection and the related data privacy and de-identification issues. Oftentimes the data originates with protected health information and needs to be de-identified before it can be shared. Increasingly, we're seeing more multimodality studies. So while we're at ISMRM talking about MRI research, we're seeing more and more projects with multimodality data, maybe various omics or EEG data, data from clinical systems such as Redcap introduced, and we'll talk a little bit about some of the challenges there and approaches for dealing with it. Dealing with differences across sites is also a common factor. If you have different scanner types and different makes and models that can impact the data, and how do you deal with those site-specific differences with issues like quality assurance and so on as well. As I mentioned earlier, Redcap integrations for collecting clinical data and kind of integrating that data back with your imaging and related non-imaging data, so you have a complete view of the subjects, can also be a challenge. So we're going to talk a little bit about how we can address some of those problems. Given that a lot of research these days is headed towards machine learning applications, an increasing use case that we see more and more of are around the ideas of multi-reader studies. How do you get radiologists involved, for example, to do reads and to annotate the objects? But these readers may be in different locations, and you need to blind the data and sample it out and deal with the whole workflow associated with that. And doing that in a distributed multi-site context has its own challenges. We'll talk a little bit about that. And then broadly, the goal typically in imaging research is to get the data back centrally located and to deal with various analytic pipelines, and then ultimately to share that and have your teams collaborate. And we'll talk a little bit about how that workflow can work both kind of in a fully automated way or in a kind of more interactive exploratory way, and then leveraging standards for sharing and so on is also very helpful. So anyway, I just want to give you a feel for some of the things that we're going to touch on today. And to get started, then we'll just kind of explain briefly what Flywheel is. So Flywheel is a software system for managing scientific research. We started at Stanford University around imaging core facility related research and data management challenges, and we've expanded quite broadly into clinical and life sciences and other AI related applications. But broadly, it's a software system that is designed to manage a research oriented workflow. So it's not a clinical tool that's VNA or something that's sort of been repurposed for research. It's really a unique research workflow. So the system helps you capture data, whether that's directly from imaging modalities or remotely uploaded. We'll talk a little more about that, but it can handle imaging and non-imaging data. We'll talk about the importance of curation. So how do you manage and organize your data, label it, classify it, quality control it, make it searchable, that sort of thing. And to help with all of that is a computational framework that can do everything from basic pre-processing to full analytic pipelines. And that can be done inside the Flywheel system itself and largely managed by Flywheel. but we also have ways to allow you to integrate and access the data and use your own external pipelines and perform various integrations like the Redcap scenario I mentioned earlier. And then everything in Flywheel is done in a secure project context, which is very well suited to the needs of really any research collaboration, but particularly well suited for multi-site research collaboration. So our customers use the product, which by the way can be deployed either in the cloud. About half of our customers are in the cloud, and or it can be deployed in your on-site infrastructure, VMware virtual machine type of scenario, if you like. So imaging research is our bread and butter. Increasingly machine learning. There's been a lot of innovation in the area of using machine learning with imaging. And then because we're web-based and secure, and many of the things we're going to focus on today have to do with multi-center studies. So I won't go through all of this in more detail, but the main point here is we have a kind of a complete end-to-end workflow for capturing, curating, and analyzing and collaborating on your research data. We now have a little over 40 leading research institution customers around the world, and most of them are running grant-funded studies or doing clinical research, or in some cases life sciences, pharma R&D. But a common theme is the idea of making data more accessible, interoperable, and reusable, kind of broadly the NIH FAIR concepts. So Flywheel does this in many different ways. We'll touch on some of these things around the importance of metadata and search. We'll talk about open APIs and tools, which are again very useful in these multi-center contexts. Interoperability, standards-based, but fundamentally it's around reusing and sharing data and allowing your various collaborators to work together effectively. And so this is a guiding set of principles for Flywheel. The Flywheel system, by the way, is very much an open science-based architecture for supporting collaborative research. So just quickly some examples. So the first example is what we're here to talk about today, which is really multi-center research collaboration. So this is a good example of a project in Flywheel. It's a Down syndrome study. Dr. Mike Yassa at UC Irvine is the data core for a project that has contributors from Columbia University and MGH, and they contribute data to the system, and then the data is kind of quality controlled and processed to a standard. Then the different teams can collaborate on that data, and then ultimately data goes back to the NIH towards the end of the process. And this project's been very successful. In fact, Dr. Yassa has expanded this solution on his site to include his own lab, and has gone on to create an entire collaborative around having loaded all of his data for his lab into his system, and then sharing that with other labs around the UC Irvine campus to create, again, a collaborative. In fact, we have a kind of new white paper outlining his use case there. If that's of interest to you, you can download that from the site. So that's a classic multi-center study, and I'm going to come back and talk a lot more in detail about the different aspects of this. But just quickly, a few other use cases. We started in imaging core facilities where you have one or more magnets contributing dedicated research scans into the users are collecting that data and need a place to archive it, to process and analyze it, and then to facilitate collaboration. And we're doing this type of project at many different institutions around the world. But essentially the Flywheel system becomes a portal for sharing data, research data that, in this case, collected as original research scans. We're doing the same type of thing increasingly in kind of the School of Medicine context, where research hospital is interested in sharing data from their PACS and VNA and imaging data, but also EMR data. Digital pathology is growing, clinical data from Redcap, that sort of thing. But broadly, the challenge is, how do I make more clinical imaging and related data available to the research community and deal with the challenges of de-identification, security, and so on, and secure collaboration. So how do I enable these users to work together across disciplines and solve new problems, including artificial intelligence development or development of precision care, personalized healthcare solutions. So we're seeing a lot of this kind of thing. And just quickly, our largest deployment is a very large pharma who is using Flywheel to ingest, very large volumes, literally millions of data sets of historical data, cleanse that data, prepare it, make it available for reuse for downstream, analysis in the context of pharma R&D, including, synthetic control arms and capture of biomarkers and so on for use in developing new solutions and treatments. So anyway, I put this one in here mainly just because the workflow is largely the same across all the systems. This one is particularly interesting from a scale point of view. But let's talk about multi-center collaboration a bit more in detail. So in the context of multi-center deployments, Flywheel looks something like this. You typically would have a core team that is the data core for the project, and they're collecting data from multiple sites. In this case, I sort of label here as project contributors. They could be contributing imaging data, non-imaging data. Typically, there's some de-identification that needs to be performed before the data is uploaded. You'll notice on the right here, we've got the concept of kind of a master project and projects per site. That's an optional construct in Flywheel, but it's very useful in the context of multi-center studies because it can help you isolate, first off, let each participating site maintain control over their own data, but you can also use it as a vehicle for quality checking and dealing with any site-specific differences that might exist between the sites before any data is either moved to or copied to the master project for the overall result. So anyway, so you have your contributors contributing data through a variety of means. I'll come back and talk a little more about that. There's lots of opportunities for automation on the back end. Quality control, labeling and classification, running pipelines. For example, we'll talk a little more about this, but in the computational context, we can do everything from running, basic DICOM to NIFTY conversions for you, or running quality control algorithms like MRI-QC, but we can also run, FreeSurfer and the human connectome pipelines, that type of full analytic pipelines for you automatically. And one big benefit is the system will track everything for you. So it's going to provide extensive documentation, provenance on everything that happened, who ran it, when it ran, success, fail, all the background details, inputs, outputs, configuration options used, everything. Can also be used to automate machine learning workflows. But ultimately, all the data is managed centrally, available for search, reuse, sharing, but all in an access-controlled, secure framework. And then, I mentioned I've discussed a lot about the background automated processing on the right here, but equally important is the ability for your teams that are doing analysis on the data to be able to easily access that data, download it, use whatever tool they want to use. Whether that's machine learning, image processing, or otherwise, and then ultimately, share that data back to the master project so the broader team, can work with that data. So this gives you a kind of a high-level feel for how Flywheel would be used in a multi-site collaboration. So to kind of hit a little more details here, a little bit more detail. The system, the first and foremost thing in a multi-site collaboration, given the diversity of users, multiple sites, different IT contexts, is to make it as easy as possible. So we start with a web-based, easy to access, but secure platform, so that it's very intuitive to log in, find your project, upload data, work with that data, and with minimal training required. We provide, in addition to just being simple and intuitive out of the box, we provide extensive videos and online documentation and everything to support that process. But the main point is you can stand up a multi-site study very, very quickly. And the tool is, easy to navigate. There are viewers built in. There's extensive tracking and logging of everything that happens automatically, just as a function of the basic capabilities of the solution. To give you a little better feel for what's really happening there is the Flywheel system can be set up to organize around your group. So if it's on site, it might be your lab. It might be a group, but if you're doing a multi-site study, you might have a virtual group just for that, that collection of related collaborators on the project. Then you can have individual projects, which could be, the master project or projects for each site, or any, individual grant funded studies. Up to you to decide how you want to use the project idea. You can have as many subjects as you need. Flywheel supports longitudinal studies very well. So there's a formal concept of a subject with many time point exams we call a session. Those sessions can be made up of any type of data that you like. It could be essentially DICOM studies, but it could also have raw data, EEG, omics, EMR data, whatever it is you need in the system. So you can kind of see at every level of the hierarchy. You can attach files, you can attach metadata, but you can also keep track of the various analyses that you've performed against the data. And on the right here, you'll see there's everything is user and permission controlled. We'll talk a little more about that in a sec. But there's also templates and rules to, you can design your study, and the system will raise flags if data is missing, that kind of thing. And we make it easy to repurpose data across by creating, new collections of data to meet your needs. So it's a research-centric system that is, designed to give you enough structure to keep you consistent, but also a great deal of flexibility in terms of being able to add metadata of any type, add files of any type, and we don't constrain you in terms of what you can do. So how would you use such a thing for multi-site collaboration? Well, the first thing is you've got to connect your users. And so Flywheel has introduced what we would call federated authentication. So this means, the ability to have you log in using your own institutional credentials, into the central site. So if you're the host of the site, what this means is that you don't have to, necessarily, unless it's a policy on your site, you wouldn't necessarily have to create, credentials at your institution, and then everybody participating, has to log in through those credentials. If you enable Flywheel to allow them to log in with their own institutional credentials, which is a configuration option, we can authenticate back with their institution, typically through a variety of mechanisms. So the federation that we use supports up to roughly about 4,000 different institutions. So if you're familiar with a service called CI Logon or In Common, many institutions participate there. Or you could use your Orc ID. EduGain is another kind of component of this solution, helping connect more institutions. We've even got the Australian Access Federation, for example. And technically, all of this is being done through a service called Auth0, which supports all the leading authentication protocols. But from your point of view, what it means is you can very quickly set up a project, invite your users from multiple sites, and they can be authenticated back to their institution's security, so that you can ensure that it's the right person logging in. And then once they do log in, they still have to have permissions granted on the project. So, just because they're in doesn't mean they actually would see anything unless they were, had permissions and roles on various projects. But the main point is we can simplify connecting the various sites for your multi-site collaboration, because we've done that work ahead of time. So then typically once you're connected, the next challenge is how do you collect data? So from remote sites, that can be quite a challenge. People are using Dropbox and Box.net, all sorts of different tools. But the problem is the data is not organized very well, and they don't really help you with de-identification. So Flywheel has introduced an array of tools for getting data into the system. So at the very simplest level, we have a web-based uploader. It's a zero footprint, nothing to install uploader, and users can just drag a series of DICOMs or a DICOM study or more studies, multiple studies in fact, into the web uploader, and it will look to see if there's a de-identification profile for that project, and if so, it will de-identify it locally in the browser before the data is actually sent to Flywheel. So simple to set up, but also helps you with the DID process on the way. For bulk loading, we have a couple of different options, command line tools and some scripting tools with that can, so you could use Python or MATLAB, for example. And then for larger collaborations where an automated connector is important, we have that option as well. So we can actually, look like a DICOM node on the local network, and the users could just push to that node and the data would get forwarded on. And of course, DID as need be. So there's a broad set of options ranging from, simple and lightweight to large scale bulk uploading to fully automated connectors, and all of those have de-identification capabilities built in. So again, for a multi-site project, many different options. Just a little bit more on the de-identification. Flywheel has fairly extensive DID tools. So all of those mechanisms I mentioned on the edge, as you're sending data in, can process our DID profile format. It's a template that configures things like which fields are whitelisted or blacklisted, can remove and replace tags, can hash values, but also can deal with dates fairly well in terms of things like, offsetting a date or computing age in years, or other ways that, essentially can convert dates into de-identified format, so you can still have the value of dates, but without, sharing any protected info. The other thing that customers can do, and this is maybe not as much relevant for a typical multi-site collaboration, but if you have the need to have PHI in some of your projects, Flywheel can do that. And if you then need to de-identify for sharing and multi-site collaboration, you can do that de-identification inside of Flywheel as well. And in fact, we can even run a quarantine service that will sort of do some extra checking for PHI and can flag and remove potential PHI if the system in the background spots anything that looks like PHI. So I think the main message here is that we can help you on the edge with your de-identification. We can help you in the system with additional de-identification, and we can also help monitor for the existence of PHI to reduce risk. Earlier I mentioned multimodality research. So again, we're here at ISMRM talking about MR research, but Flywheel is really well suited towards virtually any DICOM based processing, but also non-imaging data. So, really any file-based data that you need to collect can typically be managed in Flywheel. So these are some examples. I've mentioned some of these: physio and digital pathology, various omics, other tabular data. And regardless of whether we've seen it before or not, the nice thing about Flywheel is we have an extensible data type system. So if you have a new file type that we haven't seen before, you can essentially create your own handlers for that, both in terms of algorithms that extract the metadata, perhaps process that file type. We do that through what we call Gears, this plugin application processing mechanism. But we also have a user extensible viewer plugin API as well. So you can add your own viewers. So in this example here, I'm showing what we've done with EEG data, where we get a set of files, related files for brain vision EEG, which include a header file, the actual data itself, and then a set of markers, in a separate file that indicates where interesting events are, task events, and so on. what we were able to do then is use our GEAR framework to create a classifier that extracts the metadata, indexes that data for search, and then we were able to use our viewer plugin API to create a custom waveform viewer, for that particular set of data. And so anytime you see a new data type, we can apply this same basic model to support those new data types in the system. Kind of moving on then, I mentioned Redcap earlier. So Redcap is a system for typically collecting clinical trial data or clinical data in research projects. And oftentimes the challenge with Redcap is that it doesn't deal with imaging very well, but it might capture various forms and surveys and some clinical attributes that are of interest to you. So in a lot of our projects, we see the imaging data coming into Flywheel, but the user would like to pull in the related clinical data so that you can search and perform analyses using all of the data. And so Flywheel allows us to do that. We have, it's usually a matter of a simple script to link the Flywheel system to the Redcap system. So we have a Python-based software development kit for Flywheel that can update and read metadata easily in Flywheel. And there's a corresponding similar API for Redcap called PyCap. And so you write a simple script that pulls the data from Redcap typically and adds that custom information to the Flywheel database. So then your images or your subjects or your various exams have the properties from your Redcap database, and then you can search them, you can access them through the API, that kind of thing for writing various analytic algorithms and so on. So it's a fairly straightforward thing to do. We've seen a number of different workflows. I won't spend more time on this now, but we've had integrations where the Redcap data triggers the acquisition of the image data into Flywheel, and all the data lands in Flywheel, or we've seen the image data triggering a query back to Redcap to pull the corresponding data. So there's quite a few ways you can set this up, but it's easy to integrate Redcap and systems like Question and others that are similar. Kind of continuing the data acquisition thread here. Another increasingly common workflow is to have multi-reader studies for annotating images as an input to maybe a first step in a machine learning study, for example. So using the Flywheel multi-site and project model, we can accomplish a blind multi-reader study. So we have the ability with integrated viewers using work lists and visual ROI, annotation tools. We can set the project up so that we can sample the data out into reader projects, if you will. Those readers come in through the work list, read the, each study, essentially annotate. We do have, by the way, a configurable set of labels and a configurable qualitative summary form so that you can capture all of the annotations back to the Flywheel database, and without having to use an external spreadsheet or any technique like that. And then the data can be aggregated from the reader projects back to the master project. So you can say, okay, for each instance, what were the various reads that, for each case, what were the various reads, and did they align or did they disagree? And, then use the actual ROIs themselves to help guide your machine learning, isolate the instances that were identified and that sort of thing. So blind multi-reader studies is another kind of distributed workflow that Flywheel can help with because of its web-based nature. The now we're in the land where we have our data collected centrally. We've maybe collected it through these sites, and our goal is to get it into a master project, maybe process it in some way. So one tool that Flywheel offers here is the ability to have centralized algorithms that for anything that you want to run on a routine basis. So that could be quality control, could be format conversions like DICOM to NIFTY, could be running, your whole pipeline sequence. And Flywheel has a concept called Gears, where the tool can manage this kind of processing triggered by new data or state changes in the database. We call those gear rules. but you can also just point on demand and say, apply this algorithm to this data. And you can do that in batch as well. So you can say, here's 500 studies I want to run free surfer on, for example. And Flywheel will scale all of that up and direct that workload. You can see here on the right, it can direct it to the cloud. It can direct it to your HPC system, your high performance computing cluster at your university, running Singularity with Slurm scheduling, et cetera. But also you might have dedicated GPU boxes in your network that, for your lab, and Flywheel can direct that work down to that level as well. So Flywheel will automate the work, send it for processing wherever it needs to go. And as some of these jobs can take, 8, 10, 12 hours, depending on the nature of the algorithm. Flywheel will kind of take care of all that for you, collect the results, store it as a, kind of as provenance, if you will, and keep track of all of that processing for you. So for anything that you want to perform on a population study in a routine way, this Gears mechanism is very powerful for doing that. And we provide all sorts of tools, by the way, to help your teams build their own algorithms and plug them in. But Flywheel also has a gear exchange, a way we share with our customers. All of the Flywheel gears that we offer are delivered as open source. And there's, I think, currently a little over 100 of these in our system. And you can download those and kickstart your project. So common things like quality assurance and format conversions like DICOM to NIFTY again, and the human connectome pipeline, that sort of thing are all pre-built and are available out of the box, as are many other popular algorithms in the FSL toolkits or SPM, ANTS, MRTrix, many of those. And our customers can share algorithms in this way as well. For example, University of Pennsylvania recently introduced a new DTI pipeline called QSI Prep. It's actually a BIDSAT, and they're offering that as a gear amongst other gears they've produced. Now, gears are a very useful way to work, but that's not the only way to work in Flywheel. So if your team, say, have their own pre-existing pipeline that they would like to use, or feel like they need to do some exploratory development, you're a first-class citizen in Flywheel as well there. So we offer many ways to get data out of Flywheel, downloaded to your desktop or into your server, so that you can perform these various types of analysis. So the web UI, every project has a button, by the way, you can click to download the entire project if you want. But excuse me, any individual study or series or an individual file can easily be downloaded as well in the same way. Then you can script that either through the command line tool or through the Python, MATLAB, and R SDKs. And in fact, our DICOM interface can receive DICOMs and push them to a DICOM server if your tool or pipelines, use a DICOM server as its input. So the main point here is you can automate things in Flywheel, and Flywheel will help with a great deal of that processing, but you can very easily load up a Jupyter notebook or your MATLAB shell and just interact with the data in Flywheel and perform any exploratory development or integrations that you need to perform as part of your system. So, and basically the way all of this works is Flywheel is really very much an open science architecture, where the core services, the database, the search engine, the compute management, are all offered via an open API. Our internal connectors and command line tools and the web client all use that same interface, but we also provide language bindings for MATLAB, Python, and R through the same interface. So really everything is very open and accessible, provided that you have the permissions, you're authenticated to do so. But in terms of how you integrate your data, it's not a closed system at all. It's very, very open. And then maybe finally here, I would say standards are very important. We do a lot of work in the neuro community, and the standards there are largely driven around the BIDS standards. And Flywheel, because it's got this extensible metadata framework, is extremely useful for setting up the logic to enforce those kinds of standards. And we've gone pretty far in the way we support bids. So we have a full end-to-end bids workflow fully integrated, but the system can be adapted to other standards in different domains. But we support, in this case, importing data in bids format. Now we have an automated configuration or a curation system with configurable templates that allows you to customize how you would transform your non-bids original data into the bids standard, saving a lot of time. In fact, if you use the repronym standard at the scanner in terms of how you label your data series, the system will run pretty much fully automated out of the box. Bids, if you're familiar with bids, has a more of a class-based organization of data. So the anatomicals will be together, the functional data will be together, that sort of thing. Whereas Flywheel by default typically organizes the data by your various acquisition buckets in series of time, when you acquired it during what order of acquisition you had in a given exam. But with the bids view, we can flip that orientation into the bid-centric view, which is actually what you're seeing here on the right. So you can kind of work very natively in bids. Further, Flywheel Gears can implement bids apps. So I mentioned earlier MRI QC, similarly fMRI prep can be run as bids apps in Flywheel. So that same automated processing, scaling, documentation of the derivatives, all of that kind of thing happens as well with bids apps. In fact, we have a toolkit that makes it very easy to build bids apps and deploy them as gears. And then finally, you can, of course, export data in bids for sharing with other, projects and back to NIH and so on. Many of their sites now support bids. So anyway, so that I think is the high level what I wanted to accomplish today. I hope what you saw is a system that's easy to use, it's secure, can connect and capture any of the data that you need to capture, whether that's imaging data or related non-imaging data. We have a framework for dealing with quality and automated processing. We have tools for integration, and we have tools for doing all your exploratory development analysis, and it can be configured to support various standards that may be important to your multi-site collaboration.